Welcome back to the Deep Dive. Today we are attempting something. Well, frankly, a little arrogant. We are going to try to explain the unexplainable.
Extremely arrogant, but, I think it's necessary.
We are going to try to explain how you, yes, you, the person listening to this right now, went from being a single microscopic cell to a walking, talking, complex organism made of, what is it, roughly 30 trillion cells?
Give or take a few trillion, yeah.
And we are going to try to do it without putting you to sleep.
That is the mission. And honestly, it's a mission that requires backup. We are not doing this alone.
Right. We have pulled 3 of the absolute heavyweights off the medical school library shelf for this. We aren't just skimming a Wikipedia page. We are synthesizing the trinity of embryology.
You really are.
We have Langman's medical embryology, we have Moore and Prasad's The Developing Human, and we have IB Singh's human embryology.
It's a bit of a meeting of the minds today. And they each have their own flavor, don't they?
Yeah.
You have Moore and Prasad's, which is just wonderful for the historical context and the clinical big picture anatomy.
The storytelling aspect of it.
Exactly. Then you have Langman, which really dives deep into the molecular signaling, the software code of life, if you will.
The nuts and bolts.
And then there's IB Singh, which is this fantastic structured guide to the mechanics of how cells actually divide and organize, really exam-friendly.
Exactly. And I want to set the mission right up front for you listening. If you are a medical student, a resident, or just someone fascinated by biology, think of this deep dive as your ready reckoner.
A guide.
Yes, a guide. We are not replacing the books. Please go read the Introduction to Human Development, and Introduction to Molecular Regulation and Signaling chapters in these texts. But consider this your pre-flight briefing. We want to spark that curiosity so that when you do open the books, the concepts feel like old friends.
That's the goal, because embryology isn't just about how babies are made. That is a massive oversimplification.
It's so much more.
Embryology is the bridge. It explains why the anatomy in an adult looks the way it does. It explains pathology. It explains surgery. You cannot fully understand the adult human body if you don't understand the twisting, folding, and rotating that happen in the first 8 weeks of life.
It's the origin story for every single structure.
Yeah.
Every organ placement, every congenital defect, it all starts here.
It does.
And we're going to cover a lot of ground today. We're going to travel from, like, the ancient philosophers who thought babies came from mixing blood and seeds, all the way to the high-tech molecular switches with names like Sonic Hedgehog that actually tell your fingers to grow.
It is a wild journey. From the homunculus to the morphogen, it's quite a leap.
Okay, let's unpack this. I want to start with a history, because Moore and Prasad does such a great job of laying this out. It seems like for thousands of years, humans had absolutely no clue what was going on inside the womb.
It was a total black box. I mean, if you look at the ancient Egyptians around 3000 BC, their understanding was more spiritual and biological. They believed the soul entered the infant at birth through the placenta.
So it wasn't about the mechanics, it was about the metaphysics.
Precisely. Then you look at the Hindu texts, the Garbha Upanishad from, 1416 BC. They were actually getting closer. They had these descriptions. They said the embryo was a vesicle after 7 nights, then a spherical mass after a fortnight.
Which is surprisingly accurate for observation without a microscope. That's pretty amazing.
It is. But then you get to the Greeks. And Aristotle, who we often call the founder of embryology, he had this idea that the embryo formed from a mixture of menstrual blood and male semen.
The soil and seed concept.
That's the one. The idea was that the semen gave the form, or the soul, and the menstrual blood provided the raw material, the substance.
And that idea just stuck around for centuries.
For centuries. It wasn't until the Renaissance and the invention of the microscope that things really got interesting, and also, frankly, a little bit ridiculous.
This is my favorite part of the history. The homunculus.
The homunculus. You have to love it. So in the 17th century, you have scientists like Ham and Leeuwenhoek who finally discover sperm. They're looking through these early, crude microscopes. They see these swimming cells, and the human imagination it takes over.
It just ran with it.
They convince themselves that inside the head of the sperm was a tiny, fully formed human being.
A little man.
A little man, the homunculus. And the theory, called preformationism, was that this tiny human just needed to be planted in the uterus to grow larger. There was no development, just enlargement.
Which, if you think about it, is just absurd. It implies that the tiny man inside the sperm has his own sperm, with an even tinier man inside that.
It's turtles all the way down. An infinite regression, exactly.
So how did we get past that?
It took a long time to break that mental model. It was Caspar Friedrich Wolff in 1759 who finally said, no, look, I'm watching these chick embryos, and they start as globules. They aren't little chickens, they are building blocks.
Right.
He proposed epigenesis, the idea that development is a process of growth and differentiation from specialized cells. You are building the house from bricks. You aren't just inflating a bouncy castle that's already shaped like a house.
That is a crucial distinction. It changes everything.
Mm.
And then enter the giants of the modern era. Moore and Persaud really highlights Carl Ernst von Baer.
The father of modern embryology, and for good reason. In 1827, he identified the mammalian oozidae. But his biggest contribution, I think, was the realization that general characteristics develop before specific ones.
What do you mean by that?
He observed that an embryo looks like a vertebrate before it looks like a mammal, and it looks like a mammal before it looks human or like a chicken or a pig. We all start from a very similar-looking template.
Which leads us to the Nobel Prize winner, Hans Spemann, in 1935. This concept of induction seems to be the pivot point where embryology stopped being just observational and started being mechanistic.
Absolutely. This is a game changer. Spemann discovered that tissues talk to each other. One group of cells tells a neighboring group what to become. He called it the organizer.
So it's not pre-programmed, it's interactive.
It's totally interactive.
Yeah.
He did these incredible experiments where he'd take a specific piece of tissue from one amphibian embryo and graft it onto another. And that little piece of tissue could induce a second nervous system or a second head.
Wow.
It proved that our bodies are built through a series of molecular conversations.
I love that image. Cells having a conversation. Hey, you become a liver. Okay. We're going to get deep into how that conversation happens when we look at Langman later.
For sure.
But first, let's establish the timeline. We have this historical context, but now we need the map. Moore and Persaud and IB Singh both break down the pregnancy into distinct periods.
Right. And for you, as a learner, keeping these distinct is vital because the risks change dramatically in each period. First, you have the germinal or ovular period. This is basically the first 3 weeks. From fertilization to implantation.
This is the all-or-nothing phase, right? That's what I remember.
Largely, yes. If something goes wrong here, a major genetic error, a failure to implant, the pregnancy usually terminates, often before the woman even knows she is pregnant.
So it's a very fragile stage.
Very. But if it survives, we move into the most critical window of all, the embryonic period, weeks 3 through 8.
Why is this specific window, weeks 3 to 8, so absolutely critical?
Because this is organogenesis. This is when the body plan is being laid down. The neural tube is closing, the heart is folding, the limbs are budding out. Every major organ system gets its start here.
So the blueprint is being drawn.
The blueprint is being drawn, and the foundations are being poured. And because the cells are dividing and differentiating so rapidly, they're incredibly sensitive to outside interference. This is the window where teratogens, agents that cause birth defects, do the most damage.
And Moore and Persaud brings up the thalidomide tragedy here as a stark reminder of that.
It is the classic tragic example. You can't talk about teratology without it. In the late 50s and early 60s, thalidomide was prescribed for morning sickness.
A seemingly harmless thing.
Exactly. But it was taken exactly during this embryonic period, specifically when the limbs were forming. It disrupted blood vessel formation and cell signaling in the limb buds. The result was phocomelia infants born with hands or feet attached almost directly to the trunk like seal flippers.
Horrifying.
It proved, devastatingly, that the placenta is not a perfect barrier, and it changed drug safety and testing forever.
So weeks 3 to 8 are the danger zone for structure. Then what? Then we have the fetal period.
Right, from month 3 until birth. By now, the organs are formed, they are there. The fetal period is all about growth and maturation.
So less about building, more about refining.
That's a great way to put it. The brain gets more complex, the lungs develop surfactants so they can breathe air, the baby puts on fat. You can still have issues here, of course, but they are usually functional or growth-related rather than malformations of the body plan itself.
Before we move on from the timeline, I have to ask about this phrase that always pops up in biology classes. Ontogeny recapitulates phylogeny. IB Singh mentions this in the context of the kidney. What on earth does that actually mean?
It's a bit of a tongue twister, but the concept is beautiful. Ernst Haeckel proposed this. Ontogeny is the development of the individual organism.
My development.
Your development. Phylogeny is the evolutionary history of the species. The idea is that as a human embryo develops, it essentially replays the evolutionary tape of its ancestors.
So we go from being fish to amphibians to mammals, in a way.
Loosely speaking, yes. The theory has been refined a lot, but the core idea holds. The kidney is the perfect memory anchor for this.
How so?
We actually develop 3 different sets of kidneys in the womb. First, we build the pronephros, which is rudimentary and non-functional in us, but it's the functional kidney in primitive fish.
Wow.
Then that disappears, and we build the mesonephros, which functions like the kidney of an amphibian. And it actually works for a while in the embryo.
It's a temporary kidney.
A temporary amphibian-style kidney. Finally, that regresses, and we build the metanephros, the permanent mammalian kidney that we're all born with. We literally build and destroy 2 ancient systems before settling on the human one.
That is mind-blowing. It's like the embryo has to practice being a fish and a frog before it gets to be a human.
And it helps explain anomalies clinically. Sometimes remnants of those old systems don't disappear like they should. You can get cysts or fistulas that are essentially ghosts of our evolutionary past.
Okay, another thing that confuses people, myself included, is the language of direction. IB Singh makes a point to contrast adult anatomical terms with embryonic terms.
This trips up every single medical student, everyone. In an adult, because we stand on 2 legs, anterior means front, like your chest, and posterior means your back.
Simple enough.
But the embryo is often C-shaped or, oriented like a 4-legged animal, so we use different terms. We use ventral for the belly side and dorsal for the back side.
Like a shark's dorsal fin.
Exactly. And instead of superior, inferior, we often use cranial or rostral for the head end and caudal for the tail end.
Right, because the embryo actually has a tail for a while.
For a good while, yes. So if you were reading a scan or a text, caudal always points toward where the tail is or was. It's a much more consistent landmark in a developing embryo.
So we have the history, we have the timeline. Now we need to look at the machinery. How do we grow from 1 cell to trillions? IB Singh takes us right into the mechanics of cell division, mitosis and meiosis.
This is the engine room, and we need to distinguish them clearly because they have totally different goals.
Okay.
Mitosis is somatic cell division. It's how you heal a cut, how your liver regenerates, how you grow taller. The goal of mitosis is consistency. You want the daughter cells to be exact clones of the parent.
It's the photocopier.
It's the perfect photocopier. 46 chromosomes in, 46 chromosomes out. You have the phases everyone memorizes. Prophase, prometaphase, metaphase, anaphase, telophase. The DNA replicates in the S phase, it condenses, lines up in the middle, and gets pulled apart. 2 identical cells. Boring, but essential.
Boring but essential, I like that. But then there's meiosis. And IB Singh suggests this is where the magic happens. This is germ cell division.
Meiosis is for gametogenesis making sperm and eggs. And the goal here is not cloning. The goal is diversity and reduction.
Reduction, meaning?
You need to turn a cell with 46 chromosomes into a cell with just 23. You have to have the genetic material so that when a sperm with 23 meets an egg with 23, you get back to 46 in the zygote.
Otherwise, the chromosome number would double every generation.
Which would be a catastrophe. It's incompatible with life.
And this is where IB Singh breaks down prophase I into those 5 substages that haunt medical students. Leptotene, zygotene, pachytene, diplotene.
The mnemonics are endless. Lazy zebra pounces deftly. Whatever works. But the crucial moment, the aha moment that you really need to grasp, happens in pachytene. This is crossing over.
What exactly is crossing over?
Okay, so imagine you have a chromosome you inherited from your dad, and the matching one you got from your mom. In mitosis, you just copy them. In meiosis, specifically in pachytene, these 2 homologous chromosomes pair up so tightly that they physically swap sections of DNA.
Grade pieces.
They literally break and refuse,
Yeah.
trading pieces. They form these connection points called chiasmata. So the chromosome you pass on to your child isn't just your dad's chromosome or your mom's chromosome. It's a remix.
A unique mosaic.
It is a unique, one-of-a-kind remix that has never existed before in the history of the universe. That is the biological imperative of sexual reproduction to create endless genetic diversity.
But this complex dance can go wrong, and IB Singh highlights non-disjunction.
Yes, mechanics fail. It's a complicated process. Sometimes when the chromosomes are supposed to separate or disjoin and move to opposite poles of the cell, they stick together.
They fail to separate.
That is non-disjunction, a failure to separate.
And the result of that failure.
One gamete, one sperm or egg gets an extra chromosome and the other gets none. If that gamete with the extra chromosome gets fertilized, you have trisomy 3 copies of a chromosome instead of 2.
And the most common survivable one is trisomy 21.
Down syndrome, yes. And IB Singh also lists others like Klinefelter syndrome, which is XXY, an extra X in a male, or Turner syndrome, which is X0, a missing X in a female.
So these major syndromes are, at their core, mechanical failures.
They're mechanical failures of the spindle fibers to pull the chromosomes apart correctly during that specific moment in meiosis. It's not some mysterious curse. It's a physical, mechanical error.
Okay, that makes sense. Moving from the mechanics of the cell to the instructions that drive them. We are cracking open Langman now. This is where we get into molecular regulation and signaling. And honestly, Langman describes our DNA not just as a molecule, but as a library.
It's a great metaphor. You have about 23,000 genes. That sounds like a lot, but we used to think it was closer to 100,000. It turns out we are more efficient than we thought. But here's the thing. Every single cell in your body has the same library. Your eye cells have the gene for making stomach acid. Your liver cells have the gene for making tooth enamel.
So why doesn't my eye digest my contact lenses?
Regulation. Most of the library is locked. Langman talks about chromatin. DNA isn't just floating around, it is wrapped around these proteins called histones, like thread on a spool.
Okay.
When it's wrapped up really tight, it's called heterochromatin. That library wing is closed for business, inactive. When it uncoils, it becomes euchromatin. That's an open book, ready to be read or transcribed.
And how do we lock the books? Langman mentions a process called methylation.
Methylation is the padlock. It is the off switch. If you add a methyl group, a small chemical tag, to the promoter region of a gene, you basically silence it. Transcription factors can't bind. The gene is turned off.
And this is used for more than just turning off stomach acid genes in the eye.
Oh, absolutely. This is how X chromosome inactivation works in women. Females have 2 X chromosomes, but you only need 1 active. So in every single cell, one of the 2 X's gets heavily methylated and scrunched up into a little ball called a bar body. It's basically put into deep storage.
And there's also genomic imprinting, which Langman mentions. This blew my mind. The idea that some genes are silenced based on which parent they came from.
Yes. It's fascinating. For a small number of genes, the copy you got from your dad is methylated and silenced, and only your mom's copy works, or vice versa. It's a parent of origin stamp.
What happens if that goes wrong?
If that methylation is incorrect, or if you happen to lose the only active copy, you get very specific diseases like Prader-Willi or Angelman syndrome. It adds a whole other layer of complexity beyond just dominant and recessive genes.
So we have the library. Now we need the conversation. We talked about induction, earlier cells talking to cells. Langman breaks this down into inducers and responders.
The inducer is the talker. It's a cell that secretes a signaling protein. The responder is the listener. But Langman makes a critical point. The responder must have competence.
Competence.
It means the responder needs the right receptor to hear the signal. You can shout instructions all day, but if the other cell doesn't have its radio tuned to your frequency, if it lacks the specific receptor for that signal, nothing happens. The message is lost.
And the classic example given for this is the epithelial-mesenchymal interaction.
This is the fundamental building pattern of the body. You see it everywhere. Epithelium, which are sheets of surface cells talking to mesenchyme, the loose filler tissue underneath,
Like where?
the gut, the limbs, the kidneys, the teeth, they all form because these two tissues trade chemical signals back and forth. You grow this way, okay, now you differentiate into that. It's a constant dialogue.
Let's get into the specific signals.
Huh.
The big players, as Langman calls them, the one everyone loves to say, sonic hedgehog.
SHH. Yes. It was famously named after the video game character, which some scientists regret now because it is involved in some devastating conditions, but the name stuck.
And what does it do?
Sonic hedgehog is the master morphogen. It's involved in almost everything. It patterns the neural tube, it separates the left and right sides of the brain, it determines which finger is your pinky and which is your thumb based on its concentration.
And Langman describes the pathway, and it's surprisingly complex. SHH binds to a receptor called patched.
Right, and you have to think of patched as a bully. Normally patched inhibits another protein called smoothened. It keeps smoothened turned off.
So patched is an inhibitor.
Yes, but when Sonic Hedgehog shows up and binds to patched, it distracts the bully. It stops patched from inhibiting smoothened. Smoothened is then free to activate a cascade that leads to the GLI transcription factors, which go to the nucleus and turn on the target genes.
So it's a double negative. Sonic inhibits the inhibitor.
Exactly. It's a release of inhibition. But here's the detail in Langman that I find absolutely fascinating. The Sonic Hedgehog protein itself needs to be modified to work properly.
How?
It has cholesterol added to it.
Cholesterol, the stuff we are constantly trying to lower in our diets.
The very same. The SHH protein has a cholesterol molecule attached to its tail end. This makes it sticky and helps it associate with the cell membrane and form a proper signaling gradient.
So what happens if you have a problem with cholesterol?
Well, this is why metabolic defects in cholesterol synthesis can actually lead to severe embryonic malformations. The classic one is holoprosencephaly, where the brain doesn't split into two hemispheres because the SHH gradient required for that split never forms correctly.
That is a direct link between basic metabolism and high-level genetic signaling.
Amazing.
Okay, next pathway. Planar cell polarity, or the WNAT pathway. Langman uses the term convergent extension.
It's a great visual. Imagine a crowd of people standing loosely in a wide circle. Now imagine they all rush toward the center and squeeze together into a long, thin line. That is convergent extension.
The tissue narrows and lengthens.
Exactly. The cells move toward the midline, converge, and the whole tissue elongates, extends. This is how the neural tube lengthens. It's how the body axis grows long and tall instead of just being a wide blob.
And if this signaling pathway, the WNT pathway, is broken,
The cells don't organize correctly, the neural tube doesn't close properly, you get neural tube defects, NTDs, like spina bifida.
one more pathway, Notch. Langman calls this juxtacrine signaling.
Right, so sonic hedgehog is paracrine. It's a secreted molecule that floats a short distance. Notch is juxtacrine, meaning the cells must be touching. Direct cell-to-cell contact is required.
Like whispering in someone's ear.
Even closer, it's almost like a handshake where one person pulls the other person's arm off. Well, biologically speaking, the Notch receptor on one cell binds to a ligand, like delta or serrate, on the neighboring cell. When they bind, the Notch receptor is physically cut. It's cleaved by an enzyme.
Oh, wow.
And a piece of the receptor itself breaks off, travels directly to the nucleus, and acts as the transcription factor. There is no second messenger, no relay race inside the cell. It is a direct mechanical to chemical transduction.
That is incredibly efficient. And Langman links this to Alagille syndrome.
Yes, mutations in the Notch pathway affect tissues that need really tight, coordinated sulfate decisions. Things like the branching of blood vessels in the heart and the bile ducts in the liver. So Alagille syndrome patients have very specific heart defects and liver issues because that crucial handshake is weak.
So we have the chemistry from Langman, the mechanics from Singh, and the history from Moore. But Moore and Persaud introduces one more concept that I think brings it all together, biokinetics. This is attributed to a scientist named Blechschmidt.
Yes, Eric Blechschmidt. This is a really important shift from thinking about development as just chemistry to thinking about it as physics.
Movement creates form.
Precisely. Blechschmidt argued that genes don't just magically make a shape, genes make proteins. Those proteins change the physical properties of cells, making them wetter, stickier, stiffer, or more mobile. These physical changes create what he called metabolic fields.
Can you give me an example?
Think about the neural tube folding. It's not just that a gene says fold now, it's that the cells on what will be the bottom of the tube become wedge-shaped. The cells on the edges start pushing inward, fluid pressure builds up underneath, the tissue has to buckle. It's a physical necessity.
Like blowing up a balloon inside a box. The shape is determined by the resistance and the pressure.
That's a perfect analogy. The shape is an emergent property of the physical forces at play. Moore and Persaud mentions his terms like dilation fields where space is being made or retention fields where tissue is being held back and compacted.
I love the check-in question here. How does this change a surgeon's view of a congenital defect?
It's a paradigm shift. If you understand biokinetics, you understand that a congenital defect isn't just a missing part. It is a frozen moment of physics. If the fluid pressure wasn't right at week 4, the fold didn't happen correctly, so the tissue migrated the wrong way.
So a cleft lip, for instance.
A cleft lip is a failure of two masses of tissue to meet and fuse. Why? Because the growth forces didn't push them together hard enough or fast enough. It helps the surgeon understand the structural logic of the deformity, not just that a piece is missing.
It makes the embryo feel much more dynamic. It's a construction site, not just a 3D print being executed.
It is a construction site where the blueprints, the DNA, the workers, the proteins, and the laws of physics, the biokinetics, are all interacting in real time.
So let's try to wrap this all up. We've looked at the giants Langman, Moore, Singh. We've seen the timeline, the cell division, the molecular signals. What's the big picture here?
For me, the synthesis is this. Development is a balance. It is a balance of genetic instruction, which is the library, cellular mechanics like mitosis and meiosis, and historical constraint, the phylogeny. Langman gives us the code, Singh gives us the machinery, and Moore gives us the context and the physics.
And it really highlights the fragility of it all, doesn't it?
That's the paradox, isn't it? On one hand, the system is incredibly redundant. We have backups for our backups. But on the other hand, A single error in talk, like a failure in induction, or a single error in separation, like non-disjunction in meiosis, can alter life forever.
It is robust, yet incredibly precarious.
Exactly, and that is why we study it, not just to pass an exam, but to understand the miracle of normal. When you see how many thousands of steps have to go right from the crossing over in pituitine to the cholesterol on sonic hedgehog to the physical folding of the neural tube, it is staggering that any of us are born healthy at all.
And that leads to a final thought for you, the listener, to take away. We've talked about all these signaling pathways, induction, cells talking to cells. We usually think of this as something that happens in the womb and then it's over. But does it stop?
That is the provocative thought. We used to think embryology ended at birth, but we now know that these same signaling pathways, Notch, WNT, sonic hedgehog, they don't just disappear. They are active in adult stem cells. They are critical for tissue repair and regeneration.
And the dark side.
And terrifyingly, they're often hijacked and reactivated in cancer. A tumor is, in many ways, an embryo that never stops growing. It uses the same grow and invade signals that the embryo uses to build organs, but it does so without the off switches.
So studying embryology is actually studying the fundamental logic of life, regeneration, and even death.
100%. It's the beginning of everything.
We are going to leave you with that thought. We have barely scratched the surface of these textbooks. This was just the introduction.
The map is not the territory. Go read the books. Look at the diagrams in Langman. Read the clinical boxes in More. Memorize the charts in Singh. Use this as your launch pad.
This has been the deep dive into the blueprints of life. We will see you next time.
Keep learning.